To explain better how the parts interact, we prepared a simulation. So here we have our three operons: the invasion operon, endosomal detection operon, and cytoplasmatic operon. They are regulated by two bistable switches. The first one is here, and the second one is here. At the beginning, we don't want our system to be invasive. We don't want the cells to express any redundant proteins. We can achieve it by growing the cells on the IPTG and LACI containing medium. I will explain the process focusing on the first switch only. So let's start. The switch can be set randomly to any of the two states. Now let's see what happens when we add the IPTG. IPTG binds LACI and renders it inactive. The thermosensitive C1 represses the invasion operon, so there is no protein expression. To activate the system, we need to heat the bacterial cells to 42 degrees Celsius. Thermosensitive C1 is no longer active at this temperature. LACI is expressed, so it will repress any further expression of thermosensitive C1. GFP will be transcribed to let us know that invasion operon is transcribed correctly. Invasin will be needed for the internalization into the endosome. In the endosome, there is low pH and high concentration of divalent cations. The sterilizing will be needed later for the escape from the endosome. FOQ protein, in the presence of divalent cations, will phosphorylate the FOP protein. Phosphorylated FOP will activate the endosomal detection operon. First, the not thermosensitive C1 is expressed to switch off the invasion operon because it is no longer needed at this stage. CFP is just to inform us that everything is working. CRO protein binds the CRO box, providing additional repression and control. TET-R activates cytoplasmatic operon, which codes for another copy of TET-R. Genes after CRO box are not expressed, as long as there is CRO protein. Bacteria leaves the endosome, FOP is no longer phosphorylated, there is no expression of CRO protein. Eventually, CRO level fall down, and BAX protein and YFP can be transcribed. BAX would hopefully cause the apoptosis of mammalian cancer cells.